host you, but then he's teaching on Thursdays.
Juice,
So,
it's a juice. Yeah, juice is, I've known juice for a while.
oh, really? Okay, so maybe, ilan na ba ang nakarating?
16 na po daw. I'll play the video na po.
Sige, play mo na yung video. And then, kasi naka-record naman. Okay. So good morning, everyone in the Philippines, and good evening for those in the United States. Welcome to the PAASE webinars. As we have mentioned before, we are tuned in every other Friday morning in the Philippines and every other Thursday in the United States, so you know where to find the home base for our seminars. And so today, or tonight, or this morning, we are very proud to have with us one of our distinguished speaker, Dr. Sheree Pagsuyoin, who is an associate professor and also an associate chair in the Department of Civil and Environmental Engineering, for the Center for Pathogen Research and Training at the University of Massachusetts Lowell. So we've been trying to invite her since May, but she's a very busy in demand person. So we finally agreed that we should have this in the fall. And so with that, we are very proud to say that she is here with us now. And so, Feb, can you move the slides a couple of yeah. Okay. So by way of introduction, Dr. Sheree is an associate professor and associate chair for doctoral studies in civil engineering, civil and environmental engineering at UMass Lowell. As we were talking earlier, we're on the same side of the continent, so we're not too far from each other. Her research interests are in wastewater-based epidemiology. So you will see this is a very interesting area that a lot of people are beginning to look into. I think most of this started during the COVID. Environmental biosensors and contaminant fate and transport. She has been awarded some very distinguished recognition. She was a Fulbright Global Scholar and a recipient of a U.S. National Foundation Career Award, one of the most prestigious awards that an investigator can receive. She is an alumni of UP Diliman, where she got her BS in chemical engineering and MS in environmental engineering, and she completed her PhD in CEE at the University of Virginia. She is from Camarines Sur, to those of you who are kababayans there. And so without much further ado, please welcome Dr. Pagsuyoin. And Feb, can we have the title of her seminar? The slide with the title of her seminar. Oh, I guess you can have it. All right. So please welcome Dr. Pagsuyoin.
Thank you. Thank you very much for the very kind introductions. Just let me share my screen and let me figure out how I would do that here. Okay. All right. I think this is everyone seeing the slide in their screen?
Mm-hmm.
I think I got it wrong too. Let me start from the very top. All right, so this is the title of my talk today. So first I'd like to thank PAASE for I just recently joined PAASE actually. I was inducted in the I believe it was your R. Let me say R now. It was the
Okay.
it was the
Welcome
recent
to PAASE.
conference, yes, annual conference in UP Diliman. So I was actually online because I was in Europe at the time for my
All right.
Fulbright research. So yeah, so part of the reason why I couldn't do the talk in May was I was overseas in a different time zone. So it was really very difficult to coordinate. So I'm glad I'm here now, and thanks for the invite, and I'm happy to present this talk. So the title of my presentation is Applications of WBE or Wastewater-based Epidemiology in Long-Term Drug Use Surveillance. The work I'm presenting is funded by the National Science Foundation. Part of it is funded by NSF, and part of it is funded by the Fulbright Scholar Program. So by way of introduction, before I start with the actual discussion, I'm an associate professor in civil and environmental engineering at UMass Lowell, and I'm also the PI or the principal investigator of the Emerging Pollutants Laboratory. And the bulk of our work focuses on emerging contaminants. And so we started with drug surveillance, and then we ventured into biosensors development and water treatment, all focused around micropollutants. And so we started with drugs. I started with estrogens, and then I moved on to antibiotics. And then eventually, when I moved to Massachusetts, I started this, effort on drugs, mainly because if you are in the New England region where Massachusetts is, the important micropollutant, at least for the environmental aspect, is this drugs, is this illicit substances. And so that's what started me with WBE as well. We do a lot of different works. We do developments of methods for micropollutant detection. That's why we started with biosensors. But we also do we've started doing some work on PFAS. So the one topic that these are some of my students. We always cannot get together for some reason. So these are some of the students who have worked on this project. So that's why they are in this photo. All right. So the topic that I will cover today is one of the research research interests in my group. So, and this is wastewater-based epidemiology. So I'll cover only 3 aspects of it. So first is our work on drug use surveillance for a small university and rural town in Northeast US. And then I'll segue into in-sewer sampling, which a lot of people are getting interested with. And then I'll conclude with discussing the challenges and opportunities for WBE, specifically for drug surveillance. We do have some publications that came out of our work, and some of them I will cite during this presentation, and I'll be happy to share them at the end of the talk or later on through email if anyone is interested. All right, so first let me put some context to the research. So in the United States, drug addiction is considered a disease. So it's one of the main challenges for public health in the United States. So if you look on the top left corner of the screen, you see that in the last 10, I mean, drug use has been increasing, drug abuse has been increasing, drug overdose has been increasing as well. But specifically during the last 10, 15 years, that increase has been exponential. And in the last 2 years, around the pandemic period, we see a very huge increase, much higher than how our population is increasing. And so our overdose rates for the first time, the rates have increased to above 100,000. So this one is not normalized with population, but as I mentioned, our population is actually not increasing that dramatically. So the actual drug use per population, if this was normalized, is actually higher. And the bulk of that overdose deaths are attributed to opioids. And so there are different kinds of opioids in use in the market. Some of them are prescribed, and some of them, or the majority of them actually, are abused or illicitly purchased opioids. So it's disproportionately higher overdose deaths attributed to opioids. 78% of of overdose deaths in the US are opioids related. And there are certain parts of the US, though, that stimulants are the ones that are emerging as a concern rather than the opioids. And if you look on the bottom right corner, where the overdose rates are happening is actually not just in one corner of the country. It's actually everywhere. The orange colors that you see indicate those places or those states where the increase has been positive or upward in terms of trends between 2021 and 2022. So with respect to how we monitor drug use in the country, we use different kinds of data sources. And so one of them is emergency visits. And so if a patient is overdosing or goes somewhere, a health facility to get treated, emergencies specifically, these are typically the overdose ones, that visit to the emergency room is recorded and it gets reported somewhere. But it doesn't get recorded all the time, and it doesn't get reported all the time as well. And then one of the more common ones, actually, these are established methods. One of the other common ones is what we call face-to-face interviews or ref interviews. This is spearheaded by SAMHSA, which is a branch of the Department of Health. And so basically, if you're a random sample that's being interviewed, you get to be asked about, your drug use habits, how often do you use drugs, and what types of drug do you use, and so on. And it's a random sampling technique, but it's been in existence for many decades. So the other one is by, admission and discharges at testing centers or also at hospitals, and including substance treatment centers. And then we also have what we call prescription monitoring program, and this is voluntary. This is not mandatory for all facilities. So if I'm a doctor, and I'm prescribing opioids or even stimulants to my patient, I could actually record or report somewhere the amounts of drugs I'm prescribing, or how frequently, or what number of prescriptions I'm prescribing to all of my patients. And that data set gets collated and it gets reported on a per county basis. So these 4 methods that I've discussed are what we call traditional surveillance methods, and they're well established, and that's how we get all these different kinds of data sets. In terms of granularity, though, typically they are reported on a county basis or state basis, and it's not the same for all types of surveillance. For example, when you're talking prescription monitoring, we aggregate them by counties, but when you're talking about emergency visits, sometimes they are by county, sometimes they are by state. So there's also a pilot program that I'm aware of. This one, I believe, is led by a university in Florida. And so they call this the induce, or essentially what they do is they do different kinds of methodologies, but specifically they're targeting monitoring the black market. So they do street interviews of potential patients and they move from place to place. So they can't do it for the entire country. That's why it's called a pilot program. So they select certain places and they interview patients on the street, and they also sample patients' hair on the street. And then they also use algorithms. They use AI to look at the black market searches online, and then they try to figure out what drugs are being searched for at any given time or any given period. And then they collate the data and they report it on their website. So for all of this, with the exception of the induce, for all of these traditional methods, typically the issue is that there's different types of granularity, right? So county level is the smallest, the aggregation of data, the smallest is by county, but also the lag time can be up to 2 to 3 years. It doesn't get reported right away. You can't get all this data in 2 months, for example, just because they're collating it from different places and they want to present the data in one go. And so it's 2 to 3 years typically that a report comes out. And so if you are a health practitioner or a health decision maker, you would like data to come to you, to be known to you at a much faster time scale. So this is where we, kind of thought about, like, can we do a different approach, a non-traditional approach? And so we thought about wastewater-based epidemiology, and this was a pre-pandemic period. So with our research question really was, can we use this wastewater-based surveillance to provide a spatiotemporal profile of drug use trends in different communities? So essentially, when you talk WBE, the idea here for drugs is that when a person uses drugs, whether these are illicit or licit drugs, eventually those drugs and their metabolites will end up in the sewer system, and the drugs in the sewage would travel to the sewer through the sewer network and eventually end up at the wastewater treatment plant. And so for our study, we looked at a rural town in Northeast United States. So the demographics skews very young. So the population is actually on the smaller side. So it's 40,000, but depending on the semester, sometimes it can go as low as 20,000. So that's how much difference it can be throughout the year. So it skews young population. 65% are school age students, high school to college. So the wastewater treatment plant capacity is 7 megagallons per day. That's the design capacity, but the actual flow is between 2 to 4, depending on the time of the year. We chose this location specifically because it's almost entirely most of the town is sewered, but the sewer system is almost entirely domestic. About 97 or 98% of it is domestic, of the sewage flow is domestic. So our goal was to apply WVE to analyze this temporal pattern of drug use in this small town. So we did a 2-year biweekly composite sampling. We sampled during the weekend and during weekday. So originally our design was we would like to look at the seasonal changes. So we wanted to do a fall, spring, summer, and then the next year we'll do another fall, spring. But then somewhere during the summer, COVID happened. And so when there was a lockdown, many of the schools were closed. And so we shifted to monitoring for COVID as well. I mean, monitoring for drugs during the COVID period. So we have a long list of chemicals that we were monitoring at the time, but I only will be talking about the illicit drugs or abuse drugs. So we grouped them into two, the opioids and the stimulants. Some of you probably are familiar with some of these drugs. I don't know if you've been prescribed this in somewhere a while back, but I have for sure been prescribed one of this at least during certain periods. So we looked at a few opioids. Morphine is one, codeine is one, hydrocodone, methadone, and fentanyl. So fentanyl is the number one. I'll mention fentanyl specifically because it's the number one cause of overdose death for opioids in the United States. even if the amount of fentanyl that a typical patient would get prescribed, the potency of fentanyl is so much higher, much about I believe 10 or 80 times that of morphine, if I'm stating it correctly. And then for stimulants, we have 5 drugs that we were looking at. So cocaine is the first one. Then there's methamphetamine, which is a main component of ADHD medication, but also it's also a component of street drugs as well. And then amphetamine, and then MDMA, which is the fancy name or the fancy name for the street drugs, ecstasy. Ecstasy in particular, it has no known medical use. It is a Schedule 1 drug in the United States. And then there's also MDEA, which is the sister of, they call it the sister of ecstasy. This is called sassy in street language. So how do we collect wastewater samples for WBE? So for those who are environmental engineers, you probably are familiar with the layout of a typical wastewater treatment plant. So this here is sketched out on the left-hand side. So the gray area is what we call primary treatment, and the main treatment for pollutants happen in the secondary treatment. And of course, there's the sludge section, which is the bottom one, the green one. So where do we collect samples for wastewater epidemiology? Typically, ideally, we would like to collect the sample right at the entry of the wastewater treatment plant, let me just actually do a highlight. Okay, so which should be where my pointer is pointing to. But because we want to get composite samples, meaning to say the wastewater treatment plant gets those samples over time, or usually 24 hours, we call that composite sampling. We are confined to where the composite samplers sit in the wastewater. And that usually happens after your wastewater passes through a screen chamber, or sometimes also in a what we call it comminutor, or this where some of the solids that are very large are grounded. So right after this preliminary section right here, preliminary treatment section here, that's where the composite sampler is, and that's where our samples come from. So these are 24-hour composite samples. We take them in duplicates and we put them in amber bottles just to minimize absorption of drugs on the containers. And we transport them to our laboratory in ice. Our laboratory is about 1.5 hours away from this wastewater treatment plant. So there's really not a lot of time of travel that's involved with the transport. So in the lab, immediately we process the samples within 12 to 24 hours of receipt. So the samples are filtered through a GFC filter, and then we perform what we call solid phase extraction on the samples. And then the extracted samples are then transferred into vials, which are loaded for LCMS. All right, so once the data from the LCMS gives you a concentration data it gives you a concentration data, which you can, back calculate into the mass load of the drugs that we are looking for over the past 24 hours. And in terms of how we calculate the consumption rate for those drugs, we do use a formula that's a predetermined formula from previous sources. This one is what's being used in the European Union. So we have the concentration times the flow of the wastewater treatment plant, and times what we call this concentration factor, which it's not concentration, correction factor, CF. So that takes into account the metabolism of the drug in the body of a person, and then divided by the estimated population of the town or of the area. There are different ways by which you can estimate population. So one is using the data coming from the wastewater treatment plants, which they tell you, hey, there are X number of connections in this facility. And then you multiply that with a typical number of people in a certain household, and then you get your population served, or your estimated population. You can also get that from census data, where if you know your service area for the wastewater treatment facility, you can just get the number of people that are living in that area. And then there are also different techniques that are related to chemical analyses. So we could use the measurements for BOD or COD in the wastewater to back calculate that, or use that to estimate the population. So in this study, we used that hydrochemical parameters. We use POD to estimate population because we are aware that in this town, the number of people are changing over seasons. And so just relying on number of connections from the wastewater treatment plant data wouldn't give us that, that information of changing populations over time. All right, so this is the plot of our results. This is a historical plot over the 2 years that we were sampling the facility. So the red one on the left, these are for the opioids, and the blue ones on the right, these are for the stimulants. And the y-axis gives you the average consumption per capita consumption estimates. And you can actually see just by looking at the graph that there are changes and there are differences across drugs, and there are also changes over time. With respect to the statistical the descriptive statistics for this data sets. So again, the red box is for the opioids, and then the blue box is for the stimulants. So our median concentrations are actually wide ranging as well. We range from about a single digit nanograms per liter to 100 milligrams, single digit milligram per day per 1,000 people consumption, all the way to hundreds of milligrams per day per 1,000 people. And on the rightmost column, we also compared what a typical town or city comparable size and comparable description in the United States, what are their measurements for drug use consumption. And for some drugs, we are above the typical ranges that they've measured. And for some drugs, we are lower or just on par with them. So we were within what other towns were also measuring. All right, so actual data sets. So our goal, as I mentioned earlier, was to compare, temporal trends across drugs for this town. And so I start with seasons. So we wanted to compare seasonal changes, or we wanted to look at seasonal changes. And so the first 5 drugs, again, are the opioids, and the last 5 drugs are the stimulants. And the y-axis, we purposely put this in log scale. It probably doesn't look like it's a lot, but we purposely look at log scale here because for some drugs, the consumptions are very low, and some drugs, the consumptions are pretty high. So for seasonal, so the white one, the first lines, the clear white ones are for fall. The gray ones, the middle ones are for spring, and the dashed ones are for summer. So we see certain drugs are most highly consumed. The highest consumed ones for stimulants, it's cocaine. Where is that? Cocaine is this one right here. So the top 3 are cocaine, morphine, which is an opioid, and also amphetamine, which is a stimulant. So we also wanted to look at whether drug consumption was changing over the weekend and a weekday, especially that this one is a university town. So we wanted to look at which drugs get picked up over the weekend when there are no classes and when people maybe are more relaxed. So we found that on certain, the white one is the weekday and the gray one is the weekend. So we find that, again, so the y-axis is a log scale representation. So for weekends, we find that methadone, fentanyl, the 2 opioids, have higher consumption during the weekend, as well as cocaine, methamphetamine, and MDMA. But surprisingly, there are certain drugs that actually have higher consumption during the weekday, and that would be morphine, which is an opioid. So you see that right here. That's your morphine. And also amphetamine, which is a stimulant, which is right here as well. So we say, you note here that I don't say anything about statistical significance. So we say it's higher, but we actually note that none of these differences, they're higher, but they are not statistically higher between weekends and weekdays. So we also had a chance to look at pandemic period, right? It's not by design, it just pandemic happened. And so we're like, okay, let's continue sampling and see what's the difference between the same period before pandemic and during pandemic. And so we looked at, so the white ones are pre-pandemics and the gray ones are pandemic period. So we find that for opioids, there is an increase, a minimal increase in consumption, so about 8 to 27%, except for methadone, which did not increase at all during the pandemic. But for stimulants, we're looking at a very, very large difference in increase in consumption. So we see like it's about 74% to about 300% increase in stimulant consumption, with the exclusion of cocaine. In particular, we have a statistical significant increase for MDMA, which is our ecstasy. Oh, sorry. We also had some iron, so those are the data for the two towns. But we also were actually conducting parallel tests for the entire, for a few, not the entire, but a number of different locations in Massachusetts. And so this one is the town that we were looking at earlier. And then we also were looking at a bigger city as well. So we also did some 2-year sampling on the, 2-year city, and that's Lowell. That's where I teach, the city where I teach. And then Boston, which is a much larger town. So our interest here is to look at, differences in drug consumption in different scales. So in terms of demographic, as you can see, the town which we just looked at, it's predominantly white, and whereas Lowell is just, it has a different population. We have a significant number of significant component of our population are Hispanic and also Asians. And in Boston, we also have a very different demographics. So we have Africans, Asians, and Hispanic making up half or 50% together of the Boston population. So in addition, we also have very different educational attainment for these towns, the educational profile. So the town that we just looked at, because it's a university town, majority of the population are more highly educated compared to Lowell City or Boston. And in terms of poverty rates, though, the town that we just looked at is relatively wealthier than either Lowell City or Boston. But importantly, if you're looking at demographics or the size or the scale of the population serviced by the wastewater treatment plant, we are, so the small town had 40,000 population, whereas Lowell has 220,000, and then Boston has 2.5 million population. And in some days, it's about 3 million as well. So what's unique about Boston is that because of how large the wastewater treatment facility is, they split their wastewater influent lines into two, one which is north and one which is south. And there are also differences between the demographics of the north and south regions. So we are still in the process of, finalizing the data sets for the other cities. Well, we have finalized them, but we haven't put them together in the same format as the previous data because that previous data has already been published, but this one is still in the process of getting written out. Okay, so let me just explain this a little bit. So these are 4 data sets for methadone, which is an opioid, and then fentanyl, which is again the number 1 cause of overdose death in the United States for opioids. So that's fentanyl at the bottom. Then we have stimulant cocaine, and then methamphetamine on the right-hand side. So the blue one line at the bottom is the small town that we just looked at. And then we have Boston North and South. These are the 2 lines that are in the middle. And then Lowell, which is actually a much smaller city than Boston, is the one that's at the top. So if you're looking at the trends in terms of concentrations, these are mass loads not yet converted into consumption per capita. If you're just looking at the mass loads, you're looking at very, very distinct profiles or trends with respect to the drugs that are being consumed by the population. And one would say that, hey, if you are in Boston, you might be seeing a lot more drugs, but that's not what we actually have here. So opioids use is highest in Lowell, which is the mid-sized city. And we also note some increasing stimulant use during the pandemic period. You see that right here. So during the pandemic period, the consumption of cocaine was going higher in the cities. But in the small town, we actually did not see that. In the rural town, we did not see that trend. There are also spatial and temporal differences in drug use, even in Boston City, even if there are, the towns the communities that we were monitoring there, the ones that are in the north and the south and part of Boston, there are distinct differences between the 2 cities in terms of drug consumption. All right, so I'll switch a little bit. So I've talked about, the 2-year WBE surveillance that we did, and then I've shown you some data. I'll switch a little bit to what I've done for Fulbright. So for my Fulbright research, my Fulbright was awarded last year, and it just concluded in August, so just a month ago, a few weeks ago actually. So the goal of it was to examine the challenges and opportunities for WBE, specifically for drug surveillance in urban settings in Asia, and compare those with that of the United States, which is the data, like the data that I've just shown you, and in Europe as well. So it's a 6-month study overall in 3 countries. So I first went to South Korea in Busan, and then I went to Manila. I was hosted by De La Salle University, and then I went to, over the summer, I was in Belgium at the University of Antwerp. So, all right. So why mention that? Because when I was doing my Fulbright research, one of the topics that kept recurring was the scalability of WBE, both for COVID, which more people are familiar with, as well as for those who are doing drug surveillance using WBE. So what's this scalability? So WBE, the one aspect of it that is enticing for those who are doing traditional WBE is the ability of the wastewater-based surveillance to be scalable. So you could be monitoring buildings, and we've seen this during the pandemic, right? Like we were, sometimes you would be taking samples from the, what do you call this one, the clean out, or where the wastewater comes out of the building. We call it the clean out. So we take a wastewater sample from the clean out, so that we're monitoring a building, or you can monitor small villages. You can, in the Philippines, we call it city or blocks, or certain blocks of a community. So, and then, we could also be monitoring larger towns or cities, like what we were doing in our study. So that's the beauty of WBE that's enticing for many. And so now comes this topic of in-sewer sampling. And so most people already know that you can just go to a wastewater treatment plant and pick a sample from there, and then go back to your lab, same way that we did, go back to your lab, and then analyze it for drugs, or you can also analyze it for pathogens. So what is this in-sewer sampling? Before pandemic, this was just an idea that you can go to a manhole, open the manhole, and then get a sample from there. Same way that you would collect a sample from a wastewater treatment plant. But during the pandemic, there was a lot of interest in actually doing that for building monitoring. And so the top corner on the left was us during pandemic. So to actually do that, we needed, several people to come with us, because the manhole is not very, very easy to open. It's pretty heavy. You'd need a crowbar. You'd have X number of people helping each other to make sure that, the feet are safe when you pull out the manhole. And then the person samples, and that's me actually there and sampling from the manhole. And so we started with just lowering buckets. And so at some point we started, lowering our samples as samplers. And at the bottom, we have here our colleagues at UC Berkeley doing their in-campus sampling as well, similar to what we were doing. But as you notice here, there are 2 people at least that were doing that. So, but over the last 2 years, there's new technology that came out. And then, so instead of us opening the manhole all the time and getting samples manually in the morning or in the afternoon, there actually is new technology where you can attach your sampler, a very, very small sampler that can fit in a manhole, and you can hook it up with any other water quality probes, including sensors for water flow. So you can put them all together in a very compact encasing, and then put them in the sewer. Actually, this one it's a technology from Israel. They call it candle samplers. So it's so tiny that it can fit in a manhole that's typically only 1 foot in diameter. And so they hook it up, they leave it there for some time, usually about a week. You can leave it longer there, and it can automatically sample for you, and you can come back at a certain time period and pick up your sample. This operates by battery or sometimes by solar as well. So you collect your sample. Typically, they do it every week, because if it's more than a week, the hose that pulls the sample from the sewer gets clogged with a lot of debris. So then you cannot collect your samples at all. All right, so what are the things that are cool about in-sewer sampling? So we have new technology now, which allows for small samplers to be lowered into the sewer and to be operated automatically. But there are issues related to infrastructure. One is that, when you lower a sampler into a sewer, you actually need to pick up which sewer you're going to sample from. Like in many cities, including in the Philippines, actually, when I was doing my Fulbright research, I walked around the city, and a lot of the manholes are either on the side of the street, or sometimes closer to, closer to the middle of the street if it's a smaller street, but that there are many cars passing through. So it presents a safety issue for those who are installing the samplers, also for those who are collecting the samples from the samplers after, let's say, a period of 1 week. So it needs to be properly sited. You need to pick up a specific location for your samplers if you are doing in-sewer sampling. And also, once you picked up your sample, sometimes traffic can be an issue. And so transport of your sample can be an issue. If you are picking up samples that degrade over time, even especially in hot weather like Manila, you're talking about a sample that needs to be preserved in a specific container, let's say an ice bath or a cooler, while it's being transported to the lab where it should eventually end up in. Importantly, I've talked with so many wastewater facilities and some people who actually did in-sewer sampling during pandemic. The cost is very, very high. Imagine if you put all of this equipment on the right in a sewer, a typical wastewater treatment plant would have this. And so what it means is that every manhole that you're putting this into is being equipped like a wastewater treatment plant is. So every sewer is one wastewater treatment plant equivalent by itself. So the cost of that can become substantial, especially if you are trying to connect a network of sewers. So if you have 10 manholes, so that means you have as if you have 10 wastewater treatment plants that you equipped with a sampler. So the logistics too becomes very, very difficult sometimes. You need to have the personnel, not just the people who sample. Sometimes, often in certain locations, they would need police to accompany them all the time. And that's because they are in the middle of the street or on the side of the street where safety can be an issue. So then therefore the police would have to accompany them. And that's an extra expense to those who are doing wastewater sampling. They need to pay for the personnel. They need to pay for the police time. And then maintenance as well of the sampler. As I mentioned earlier, debris can get caught up in the sampler. Even if you just clean it for one day, the next day some big thing comes up in the sewer and it gets suctioned off by your sampler. And so you need to come back and remove that manually. And then importantly, I think from my Fulbright research, a lot of people that I've interviewed, the medical personnel, those who do WBE, those who do wastewater collection, really the one issue that's always been raised by every one of them actually was this population scale. So how big of a population can be sampled? So as you know, sewers can be, manholes, you can pick them such that they capture the sewage flow from a specific number of people within a community. And so how small is small? In Europe, they set a criteria for how small a community that's monitored would be. And I believe it's like, off the top of my head, I can't quite recall this exact number. It's probably around 5,000 or 10,000. That's the minimum. And so any smaller than that, they would not want to install a sampler in a manhole. All right, so one example of in-sewer sampling that's still ongoing actually is being done in Boston. I'm not involved with this, but I am familiar with the people who are doing this. So this was started during the pandemic, and the data that you are seeing here is the weekly data that they post online coming from here. So for COVID, the data usually is available 3 days after the sample is taken, or sometimes a week after the sample is taken. So this is Boston City, and they have this is smaller. By the way, this is not the same Boston that I've looked at earlier. So the data that I've shown for drugs earlier, it's representative of about 3 million people. Whereas this side here is just for Boston City. Whereas the Boston that I was referring to earlier, it includes the metropolitan Boston area. So this is just for the smaller fraction, which is the city. So they put 11 samplers in manholes. Since this one has been going on since 2022. They started with COVID. The data here is for COVID. But then very recently, they've pivoted this program to also be looking at drugs in the sewer as well. So the size of the manhole, the population served by the manhole can be as low as about almost 1,000, I believe, or a little over 1,000, and then up to like 5 or 20,000. So the size of the circle that you see here represents the service area of the population and the service area of this town. So, all right. So to sum up, so our work our 2-year study on WBE, we have demonstrated the application of WBE for monitoring drug use in a town. We've tracked seasonal changes, seasonal drug use trends, as well as weekday trends, as well as academic cycle trends. We note the high use of stimulants in the town that we monitored. We also note the changes in drug use trends during the pandemic. But within the region, within the larger region where we looked at the small town, the medium-sized city, as well as the metropolitan Boston area, we know that drug use trends differ even among adjacent communities, and that these trends typically would reflect data from the surveys. So for in-sewer sampling, the most dominant issues would be the very high cost of sampling, and then of course, and the ethics as well of the ethics as well with respect to the design of how big of a sampling area or a service area the monitoring is being done on. And then for drug use, I would note that some of the people that I've interviewed who are interested in WBE mentioned that we don't need to actually monitor drug use every day or every week like you would in COVID surveillance, because drug use doesn't change drastically daily. But certain people would also argue that in some of those daily samplings that they've done, they actually would note sometimes a spike on a daily basis. And so it's kind of like two different area mindsets about how frequently you should sample for drug surveillance. But at the end of it, though, what they make an argument for is the value of this data. Should you be doing in-sewer sampling or not, depending on what is the use of the data. If you're monitoring a town that's very tiny, or if you're monitoring an area which you think is very critical with respect to drug use, then an in-sewer sampling might be needed. But overall, in-sewer sampling is not as critical for a larger city. So now moving on to what are the challenges and opportunities for WBE. So WBE, I think the public came to know more of WBE, not because of drug use through WBE, but because of its application to COVID. But WBE actually has been in existence for almost 2 decades now. Initially, we were doing that for drugs, and then it moved on to other prescription medication that are not controlled illicit drugs. But then when pandemic happened, the focus was on its use for COVID surveillance. It used to be that when we were going out to ask wastewater treatment plants for samples, and when we tell them that we are going to monitor or be looking for drugs in your wastewater, sometimes you would get a no, a hard no, even if it's a public utility, a hard no, like, oh no, we can't give you samples every time, and so on and so forth, especially when we say that we are looking for drugs. They worry that we would use the data and publish it, and then it would cause some negative press for the town. But during pandemic, though, there's a switch. There's a switch in mindset where we actually get calls from wastewater utilities, and they ask us, "Can you analyze our wastewater for COVID biomarkers, and can you tell us what's the viral load in our wastewater?" It's a very different mindset. So that being said, because so much has been done for WBE in the pandemic era, for COVID, there are a lot of lessons that we can learn from WBE for COVID that can be translated back to WBE for drug surveillance. So the first one is what we call standardization of protocols. So in the United States, we have a national level reporting of data for COVID. And because it's national level, the CDC wanted to have a standardized way by which data or samples are actually processed. And so they came up with protocols for standardization of all different samples that are coming to different labs. In COVID surveillance for drug use, there's also a standardization of protocol, but it's mostly in Europe. So Europe has what they call SCORE network, and their goal is to standardize the sampling for WBE for drug surveillance. So it's very, very similar to what they do for COVID. In the United States, we don't currently have that, but we have some institutions in the United States, some universities that work on WBE that started to participate in the SCORE harmonization in Europe. in the standardization of protocols in Europe. Our lab is hoping to join that core network as well in Europe by next year. So we would also standardize our methods with the European protocols. So, voluntary WWTV participation nowadays is very, very easy. And I would say not very easy. It's easier, a lot easier to get samples at the wastewater treatment plant and to be getting them very frequently for drug surveillance. Centralizing data access and visualization is one thing that COVID has done, but WBE for drug surveillance has not yet been able to do. There are certain cities, certain towns that I'm aware of that publish their data for the city that they are monitoring. But for those different cities that do WBE for drug surveillance, including mine, we actually don't. All right, so challenges for implementation, it's the ethical issues, stigma for drug use is different, and then we also have scale of sampling, as well as at the end of it, who will manage the surveillance, right? Like who is looking at this data. Importantly, for drug use, our main issue, our main problem is the funding stream. When you look at COVID, the data, the COVID research progressed very, very fast across different regions just because of the infusion of a lot of funding in the billions of dollars in total. But for drug surveillance, we don't have that amount. You'd be very lucky to actually get a few hundred thousands just to establish a monitoring program. So that is something that we have not been actually able to, shed light on, or like encourage our agencies here, our funding agencies to fund more work on WBA for drug surveillance. I skipped this one, and I'd like to thank the funding agencies, the NSF, Fulbright, and also our UMass Lowell Research Office. And if you have questions, I'd be happy to answer them. Sorry, I didn't keep track of the time very well.
Thank you very much. That was very interesting. So at this point, I would like to acknowledge two of our co-lectureship awardees, Dr. Ann Villalobos and also Dr. Diana Aga, who also does research with water.
Oh, she's here. She's here. I'm happy to hear that she's here. One of the pioneers of WB in the United States.
Yes.
Way before my time.
All right. So at this point, we would like to open this to questions. Anybody has questions, put it in the chat box, or you can actually just open your camera and ask questions from our guest speaker. So very interesting studies. I have one question. With the drug surveillance, are law enforcement officers interested in utilizing this?
Good question. It depends on who you ask. So, it's kind of a controversial topic, and it's because it's recorded. I will be very careful with how I answer it.
Okay, all right. We might get you into trouble.
So it depends. Let me just stop sharing. I think I haven't stopped sharing yet. Can the moderator stop my screen, please?
Feb.
Thank you.
Okay.
All right. So law enforcement, right? When we do WBE, we typically work with the Department of Public Health as much as we can. And their one concern is the data that you're going to be shared with law enforcement. And sort of sometimes that drives the conversation. And if you actually say yes, then they're very hesitant to work with you, because that's not the purpose of what they wanted to get the drug use data for. And so for some cases, like in Australia, it's used for police work to monitor where the drugs are. So they actually do WB for that. In the United States, we don't currently do that. In Europe, from my interviews, they also try not to do that as well. So if it's being used for police work, they're not that receptive. Yes, that just so that's kind of, I guess, blanket statement that I could share.
Yeah. I heard once that one of the concerns, particularly with the water samples, is that if you go into smaller and smaller areas of sampling, you can actually identify individuals based on, for instance, genetic data and the things that they shed that can be tracked genetically. So it was that kind of so a lot of people do not want to be involved in such kinds of studies to protect the privacy of the people that are providing samples. Billy, I also welcome my co-host, Billy Cabillon. Do you have a question? You always have insightful. You always have insightful questions.
I'm always being asked to ask a question. So therefore, I will ask a question. So I'm interested in your foray to the land of the morning, the country of my birth, the Philippines. So you have been there for 6 months. And I had the same question as what our vice president has asked. And you mentioned about usability. Usability is the main challenge there. In the Philippines, then, if you deploy such an expensive, project, uh, of what use is it going to be? And I underline this because, naku, manood ka sa television ng Pilipinas, yung drug killing and all of that, ano? So, how do you know that the epidemic in the Philippines of drug use is down? Can you use this technology? And in the light of Dr. Marge Peña, can you pinpoint exactly where in the Philippines are methamphetamine being produced and where they are used?
That's a very, very interesting question that I've been actually asked that as well when I was in the Philippines. So I was in the Philippines for 3 months. And I was there after I was there this summer, so it's after the whole, the era of the previous administration has ended. Let's put it that way. So that said, can WBE pinpoint where these drugs are being produced? The quick answer is no. We cannot tell where they are produced. We can tell where they are being consumed in high levels. But in the Philippines in particular, one of the challenges is the sewer system. So the estimation for WBE is based on the idea that you have an idea of the flow within the sewer area. And in the Philippines, even in Manila, where you have sewer networks, not 100% of Manila is sewered. And in some cases, you can't actually tell which areas are being captured by the network. And so that's the one challenge that I've found from the research that I've done over the summer. So another one is, the sewer inflows in Manila can be very high just because of how old our pipe networks are over there. I know that they're renovating it, but that's also a challenge for the estimation. And I think the other one also that I note is the usability. You mentioned about usability, like who's gonna use the data? Part of my Fulbright research was doing a lot of different interviews with different potential users of WB, including medical personnel, including those who are involved with drug surveillance. So I interviewed people from different parts of the world, not just the Philippines. And so one of the things that were said for the Philippines, for the Philippine interviewees was that we can't even afford the traditional surveillance and making sure that we have all the data every year or every month for the traditional surveillance, which is like a survey-based or getting the data from the hospital. We can't imagine how we would spend more money or, reallocate the money towards WBE. So that is one of the challenges that we identify if WBE will be done in the Philippines. How usable is it when on the get-go you can't actually correlate that or in parallel with traditional surveillance because you don't even have it. But I'd say it's also actually an opportunity, right? Because you don't know what drugs are being used, but this profiling can tell you exactly what the panel is. Even drugs that you were not looking for, let's say ketamine, for instance, and tramadol are increasing in consumption everywhere. I don't know if it's increasing in the Philippines. You can pick that up from just looking at the profile of the sewer.
Fascinating. Yeah. So how widely used is this in the United States? I mean,
WBE, if you are talking about WBE for COVID, it's used everywhere. It's our data
yeah,
set is the largest in the world, right?
it's in
But
here.
for drug surveillance, it's in different regions. Like in the New York area, you have the group of Dr. Aga, you have me on Massachusetts side. I believe there's also one from SUNY. I think SUNY is also in the New York area. And then we have some people in the southern part and in the middle part. Well, there's Arizona for sure. You mentioned that earlier. And then there's also a researcher that's very active in it in Washington State. So it's not a lot. It's not every
Mhm.
state, but there are certain people who do that, who have been doing that for a while. Our challenge really is the funding. Even if we come
Mhm.
together, we can't, you know, get enough excitement from the CDC to get funding for such a large scale study. There has been very few studies that have been funded, but the scale of funding for COVID is very, very different with the scale of funding for drugs.
Are they monitoring other diseases apart from COVID based on this? Like when the flu season
Mhm.
comes in, do they do that kind of monitoring as well?
You can, and there are people that have started that, but more of like an academic research. In Boston,
Mhm.
though, I would have to say, um, there is a pilot study, and that's for the sewer-based sampling that they did. So they're doing a pilot study now where they're using the drugs, but they're also monitoring two types, I believe, of infectious diseases. I forgot what it is. It's not influenza, but it's an infectious disease. So it's a pathogen that they're monitoring side by side with the drugs, and it's in the same sample. They're taking the same sample from the sewer that's being used for drug analysis. So they're all parallel studies, and they think that if it's successful, because it's an investment to sample, like to give you a scale of how much it costs to sample from a sewer over 6 months, it's about 50,000 per sewer, US dollars, just to pull up a sample, not to analyze the sample, to pull up a sample. So,
Okay.
and that's the cheap one. They can go higher. So it's very big. You convert that to pesos in the Philippines, and you toast the entire budget of the wastewater facility in just one manhole.
But you talk about Europe. So in Europe, is it more commonplace then, since they're standardizing it already?
So Europe is unique, right? Because you can think of them as very small cities because,
Yeah,
1 million
yes, yeah.
sounds like one United States. And so because there are so many places in Europe that they're sampling for and monitoring, and they do that all at the same time within a specific period. So they have data every year for a lot of drugs. This is the core network that I was referring to. So yes, they're doing that in many areas of Europe, the entire country, because it's so tiny, it's completely sewered, like
Yeah.
Amsterdam completely sewered. Belgium is highly sewered. And sampling is so easy. When I was in Antwerp, you drive 20 minutes and another 20 minutes, you cover the entire country. There's no such a thing. I drive the entire day and I'm only covering parts of Massachusetts. So it's a very different scale. But they're doing that in Europe at the wastewater level, not really at the sewer level, because the sizes of their facilities are much, smaller than us. For them, like 1 million is very big. But for us, like Boston, 3 million is kind of big, but not very big. New York is bigger. Washington, D.C. wastewater treatment plant is a lot bigger. So the scale really is very different. Um, Another one is I know that they have done some in-sewer sampling for COVID as well. It's a pilot study more of rather than a long-term one. And that one was done, I believe, in Louisville in Belgium, I think. And I haven't spoken with the person for that, but they've done that as well, like a more like a study only, it's a university that did it though.
I think we did that in our university during COVID and identified which dorms had high rates and just shut them
Correct,
down.
correct,
Those were the
correct.
days when you can do that. Yeah, instead of testing every single person, they sampled the waters and were able to find where the highest rates of COVID were on campus. Yeah.
And dorms can have 200 people sometimes. Like one of our
Yeah.
dorms are like 200. I'm like, oh, is that okay? And because I've known about the ethics issue even before for the
Right. Yeah.
drug stuff. And then I was being
A
asked,
lot
like,
of
can you
ethics
sample
issues
some?
went out with COVID, but I guess now
Right,
we
but like,
can't.
can you sample a dorm? I'm like, yeah, technically you can sample every floor, but I'm like, I'm not doing it. So yeah,
Yeah.
I don't want to do that.
Do we have any other questions from the audience? This is a pretty fascinating talk. Yeah. I've been hearing more and more about this water-based epidemic. Okay. All right. I don't see any other questions, so I really appreciate this for coming for giving this lecture today and taking the time to do it. Feb, we have a certificate of appreciation that I'm gonna read for you. Feb.
Oh, we have a fancy certificate. Thank you.
Meron tayong certificate palagi. Okay. And then we will email this to you so meron kang kopya.
It's okay. Thank you.
All right. Certificate of Appreciation is presented to Sheree Pagsuyoin, PhD, for being our speaker at the Philippine American Academy of Science and Engineering, PAASE, webinar titled Applications of Wastewater-based Epidemiology for Long-Term Drug Use Surveillance, held on September 20, 2024. Presented this day of September 20 with our deepest gratitude. And this is signed by our president, Dr. Gladys Completo, who unfortunately could not attend today because she had several meetings, but she was looking forward to it, and I'm sure she will listen to the recording of this. And so please, everybody, join me in thanking Dr. Pagsuyoin, and we hope to see you again soon. The APAMS meeting will be here in the US, so we hope to see you and bring all this. We actually have a water institute here at the University of South Carolina, and I'm sure they will be very happy to hear your research. And so thank you again, Sheree, and I hope to meet you in
Thank you.
someday soon.
Soon.
Okay.
You're on the same side of the
Yeah,
planet.
no, pwede tayo mag-drive. Okay, there you go. And so I would like to announce at this point our next seminar. Billy, I think you're one of the hosts for this, and this will be presented by Dr. Manuel Vidal Jr. This will be on October 18th. It's a Friday. Well, Friday in the Philippines and Thursday here. And it will be on the development of a placenta on a chip model to evaluate the effects of endocrine disrupting compounds in inducing acute placental dysfunction. So another very interesting seminar coming up. So everybody's invited to that, and we hope to see you again soon, and watch out for the announcements for the seminars. Okay, so I guess give a big hand to Sheree, and you
Thank
can
you,
react on that one. And thank you so much again, everyone, and see you next week. Yeah, and
Thank you. Thank you, Po. Good night.
thank you,
Good morning.
Sheree.
Thank you, Doc
Okay.
She.
Thank you.
And thank you, Feb, for all the background work. Okay.